Hello biologists, welcome back. We're going to be talking about meiosis today, which is sexual reproduction on a cellular level. And this has a lot of similarities to what happens in mitosis, but there are a few very important differences as well. So this entire lecture is really going to be all about comparing and contrasting meiosis to mitosis. Okay, so let's go ahead and start off with this question. Why have sex? What is the importance of having sex? What is the advantage? And I've got a couple of choices here, and I'll let you kind of look at those. It's because everybody's doing it, or because it's super fun, or because we're bad people, or maybe it's just to make babies. But better yet, how about this? This is really the correct answer. Sex is important because it produces genetic variation among offspring, which aids in the survival of the species. So here you had your own opinion of sex, and it turns out that it actually has a very important biological significance. Okay, so what is sex really all about on a cellular level? It is really all about offspring inheriting a new combination of DNA that leads to a greater amount of variation in traits within the offspring. Now, in contrasting this to mitosis, in mitosis there's also a DNA outcome to this, where the two daughter cells are inheriting the DNA from the parent cell. However, in meiosis, the outcome is genetic variation instead of exact photocopies of the original parent cell. So, for example, this is the opposite of sexual reproduction, is asexual reproduction. We had mentioned this before, that anytime you see an A in front of a scientific word, it usually means not having it or not possessing it. So asexual reproduction means not including sex. Now, the advantage of asexual reproduction is that an individual can give rise to genetic clones of itself that are identical, and it can do this rather rapidly. So in asexual reproduction, you can produce lots and lots and lots of offspring very quickly. In sexual reproduction, though, you can actually produce variation. And the outcome of two individuals that are genetically different and maybe even look distinctively different can produce lots and lots of offspring. Now, you know this to be true. Think about yourself with your parents and with your brothers and sisters, and if you have children, think about your children. You may look very similar to your brothers and sisters. You probably look very similar to both your parents, if not one or the other. And if you have children, your children probably look similar to you too. But one thing's for sure, they don't look identical. Now, the only caveat to this are for those of you that are identical twins out there, but that's caused by a completely different thing. In pretty much every case except for identical twins, you look similar to your relatives, but not identical. And that's because you are a unique genetic combination of your parents. You are a genetic combination that has never occurred on the planet in the past. So this is actually very important, and this is what has led to this happy family right here. So, sexual reproduction. Sex involves three major things: meiosis, which is the topic of this lecture; gamete production, that is the formation of sperm and eggs; and then fertilization. Now, we're not going to cover fertilization here, although it is certainly very important. Gamete production we're going to touch on, but really the bulk of this is all about meiosis. So let's start with a few things that you probably already know, or that we have mentioned already in the past. You have chromosomes that came from your parents. You have 46 chromosomes, right? Of course, there's not actually 46 individual chromosomes. You actually have 23 chromosomes. And the 46 chromosomes that make you up, 23 of those came from your mother, and 23 came from your father. That is, they came from the eggs and the sperm of your parents. So that's where you get the total of 46. Now, we brought up the analogy last time when we were talking about mitosis of an encyclopedia. Think about your chromosomes as an encyclopedia on how to make you. In our typical, like, Encyclopedia Britannica, when we used to have those bound volumes that we kept on our shelves, essentially every letter had its own encyclopedia. So if you wanted to know all about aardvarks or apples or airplanes, you would go to the A encyclopedia and the information would be there. What we're saying here is that you would essentially get the A encyclopedia from your mother and also a copy of the A encyclopedia from your father. Okay, you get one from each of your parents, and the information in them is going to be very, very similar, but not identical. Okay, think of it like this: is that if you had two cookbooks and you turned to how to make mashed potatoes in one cookbook versus the other, the instructions may be slightly different, although they are certainly both instructions on how to make mashed potatoes. Okay, so the variation, what you might get out of that, would be slightly different tasting mashed potatoes, but they were still mashed potatoes nonetheless. Same thing here. If you think about something that makes you up, let's say your eye colors or your skin color or how tall you're going to be, the information on how to make your eyes came from the encyclopedia from your mother and also the encyclopedia from your father. It's a combination of the two. So we'll talk more about that later. What you're looking at here is something called a karyotype. A karyotype is where the cell in your body has been completely broken open and only the DNA has been gathered. And what they've done here is they've actually lined up the DNA right next to each other, all 23 pairs. So here's pair number 1. There's the DNA from mom, there's the DNA from dad. Here's chromosome number 2, DNA from mom, DNA from dad, etc. All the way around, all the way down. And each one of these chromosomes has a pair. Only in one instance do those pairs not normally look alike. Right here. This is chromosome number 23. And I happen to know that this came from a male, and we know that because males have one X chromosome right here and one Y chromosome. In females, this would be identical. Females have two X chromosomes that are of the same length and contain the same sort of information, although certainly aren't identical in their DNA complement, but at least they are very similar to each other. But that's not true in males. Males only have one X chromosome and they have one Y chromosome. This is in fact what makes us male. Okay, so that's a karyotype. How do we get to 23 chromosomes? I want to back up really quickly here, and before we discuss this slide, I want you to think about mitosis. In mitosis, you started out with 23 pairs of chromosomes. When you were done doing mitosis, you had 2 cells, and each one still had 23 pairs of chromosomes. Said another way, each cell in your body contains 46 total chromosomes. That's where I get 23 pair. And when you're done with mitosis, there are 46 chromosomes still in those offspring daughter cells. Now, how does that compare to meiosis? Well, in meiosis, what you are doing is making sperm and eggs. You are making cells that have half the complement of DNA. So instead of having 23 pairs of chromosomes in your body when you're making sperm and eggs, you are going through a process, meiosis, that cuts that complement in half. So each sperm and each egg in your body only has 23 individual chromosomes. Not 23 pair, not 46, just 23 individual chromosomes. So meiosis is really all about how do we get to that stage. Okay? So at some point in the meiosis process, we have to go through some sort of a reduction stage or a reduction step. And so we're going to cover that. And how does that happen? Well, here's where it happens. Again, it happens in the ovaries and the testes of males and females. And in every cell of your body except for your sperm and eggs, every one of these other cells here, you have 2 pairs of DNA. You have 23 pairs of chromosomes in your skin cells, in your liver cells, etc. The term we use for that is diploid. Diploid. Di, or di, means 2. Ploidy refers to the complement of DNA. So diploid organisms have 2 pairs of DNA, and we refer to that as being 2N. That's every cell in your body is diploid, except for your sperm and egg cells. So in your ovaries and in the testes, meiosis goes on, and in meiosis, the cells in here essentially have their DNA cut in half in the process of cell division. And so you end up with sperm and eggs. Sperm and eggs have only one complement of DNA, not 2 pairs of chromosomes, but one set of chromosomes. And the term we give to that is haploid. Haploid cells in humans only occur in sperm and eggs. So those are your haploid cells. When a sperm and an egg get together, that is when the sperm fuses with the egg, voila, you are back to being 2N again. You're diploid again. Because you have your 23 that came from the sperm, 23 that came from the egg, and they have formed a diploid zygote. A zygote is a fertilized egg at that point. And then this cell right here divides and divides and divides until it becomes a baby. The baby then grows up to become an adult, and the adult at that point then begins making sperm and eggs in their ovary and testes again, undergoing meiosis, and the whole thing repeats itself. So that's the process there. Now, if that's the case, here's another karyotype I'm showing you. Why don't we just split the DNA up? Why don't we just cut it right down the middle and take this chromosome and give it to one sperm, and take this chromosome and give it to another sperm? Wouldn't that also equal 23 pairs of, or wouldn't we also be cutting it in half and getting 23 chromosomes into the sperm and the egg that way? Wouldn't that work? Well, there's a problem with this. You see, the whole purpose of sex is to try and create genetic diversity. And if you were just to split those chromosomes up like this, let's say that here's the one that you got from your mother, here's the one that you got from your father, and now all you're going to do is divide them up so that you're going to split them like this, well, that won't work. Because it means that all of the genes, all of the genetic material from one chromosome would go into one sperm, while all of the DNA from this chromosome would go into another sperm. And we need to include a step here that actually shuffles that genetic material to produce that unique genetic makeup that the planet has never seen before. So sexual reproduction actually shuffles the alleles. It doesn't just split things down the middle, it actually shuffles it. And so what this means is that offspring, your babies, are going to inherit a brand new combination of alleles, a combination that came from you and your spouse, just as you are this brand new genetic combination of alleles that came from your mom and dad. And this, by the way, is going to be a very important point for the future when we talk about how evolution works. You see, evolution works on the principle that in succeeding generations, there is genetic variation that can be inherited. So we'll come back to this point a little bit later. Okay, just a reminder, where does this happen? Well, we already pointed out that it happens in the testes and the ovaries of males and females, but the same thing is going on in flowering plants as well. So this is a flower showing you the anthers here, and this is the equivalent of the male gametes, or it would be equivalent to our sperm. And then over here in the ovules is the ovary where eggs are being made, and that would be the equivalent to like the ovaries of a female producing the eggs. Okay? Now, meiosis differs from mitosis in that there are actually two sets of cellular division, two sets. Fortunately, they use the same names, and they just refer to them as phase 1 and phase 2. So we're going to go through each step, and I guarantee you're going to have to go back through this and watch some of the videos I have online to help kind of sear this into your brain. It is kind of tedious, and you're not going to get it the first time. So don't get frustrated. You're going to want to come back and watch those videos a number of times. The first step is meiosis 1, and in meiosis 1 we've got prophase 1, metaphase 1, anaphase 1, and telophase 1. We're going to go through each of those steps in just a second. Once that is done and the first set of nuclear divisions has taken place, you then go through the second set of nuclear division, called meiosis 2. So that includes prophase 2, metaphase 2, anaphase 2, and telophase 2. So fortunately you are already familiar with these names. And so what you end up with here in these two nuclear divisions, in meiosis 1 and meiosis 2, you end up with 4 haploid cells. Now, let's start at the very beginning here. Here's the 2N cells. Remember, this is the cell that has two pairs of DNA. It has your 2N here. And this is occurring in ovary cells and in testes cells. Not in the sperm and eggs, but in the ovary and testes cells that produce the sperm and eggs. So your 2N here, just like the rest of the body. And just like in interphase, in the S phase of mitosis, at some point there's also an S phase where the DNA duplicates itself, just like in mitosis. Okay, but this time we're doing it only in the sex cells. So during this duplication, you now have your sister chromatids back. Oh, hey, we remember sister chromatids, right? These are photocopies. They're genetic complements of each other. Here is sister chromatid from mom, and here's the sister chromatid that came from dad, these two sister chromatids. The first meiotic division, so in meiosis 1, we're going to take this and simply split these two chromosomes apart. After we've split them apart, we're then going to go through meiosis 2, which then pulls these two opposite poles and produces 4 1N cells. So essentially, one testis cell makes 4 sperm cells. One ovary cell makes 4 egg cells. Okay? Now let's move on, or actually what I want you to do here is, at this point you can pause it and take a look at this meiosis overview video. This will be really helpful for you. So this is now before all of the meiosis stages go on. We're in interphase here, just like in mitosis. During interphase, in the S phase in interphase, is when the chromosomes replicate themselves, yielding 2 sister chromatids that are essentially photocopies of each other. So this side over here is the exact photocopy of this side here. They contain the exact information. The same thing over here. This side is the exact information as this side over here. Okay, you remember that. However, in meiosis, something really curious happens. After the DNA have duplicated themselves, and the homologous chromosomes are now set right next to each other. This is the one from the mom, and this is the one from the dad. Something really weird happens, kind of weird and wonderful at the same time. And by the way, remember, each of these chromosomes here, each one of the pairs of chromosomes, is considered a homolog, right? Now, the thing that is really weird that happens is that the non-sister chromosomes, that is like this strand right here that came from your mom, and this side over here that came from your dad, actually begin to exchange little tiny segments of DNA. And this illustration here is showing you this little fragment of DNA here that is actually switched. It's kind of weird. Isn't that weird? I don't know. I kind of think it's weird. So each chromosome then attaches to a spindle and gets pulled apart. But when that happens, I want you to see what ends up going into the 4 daughter cells at the very end. Okay? So jump all the way to the end. This act of crossing over, of exchanging genetic material here, creates 1, 2, 3, 4 completely unique chromosomes. Completely unique. Remember, these two up here were identical to each other when they duplicated each other. But when they crossed over and genes were exchanged, DNA was exchanged here, it now means that this chromosome is no longer identical to this one. And this chromosome is no longer identical to this one. So interesting, crossing over has resulted in 4 unique genetic combinations. So what is the effect of crossing over? Well, each chromosome ends up containing still both the maternal and paternal segments. So there's still the new DNA strand that you just made. It still consists of mom and dad DNA. It's just now, instead of those being separate on separate chromosomes, we've now mixed them together. And that last picture back here implies, this picture kind of made it seem like only one fragment of DNA is changing over here. But no, in the real world, it looks more like this, where whole sections, little tiny fragments, hundreds of them, all up and down the chain, end up recombining. And so crossing over is incredibly important. It is the first step that leads to genetic variation in an offspring. And there are actually a number of other steps that we're going to talk about coming up here that also contribute to genetic variation. This is the first one of them. Okay, so it creates new allele combinations in offspring. Now, after we've crossed over, right, we're done. We're done with the crossing over bit now. And this occurs during that interphase stage. Now let's jump to metaphase. In metaphase, remember in metaphase and mitosis, all of those chromosomes lined up on that equator. But in metaphase 1, it's a little bit different. It is the homologous chromosomes that line up in opposition to each other along that equator. Okay? So the chromosomes all move to the middle, and one homologous chromosome orients towards one pole, while its complementary homologous chromosome orients to the other pole. And these spindles begin to fully form. Okay, they attach themselves to the kinetochores in the middle, and then they pull apart. They begin to pull apart. And just as in mitosis, in meiosis, they pull apart in something called anaphase. But this is anaphase 1. We're just pulling the homologous chromosomes apart. And the sister chromatids, by the way, are still kind of sticking together, right? So they haven't been pulled apart yet. Notice, though, that the sister chromatids are not identical anymore, right? Because they've done a little bit of crossing over in the process. Then we enter telophase 1. After they've been kind of pulled to those poles, the nuclear membrane begins to reform, and the cytoplasm then begins to divide, and you end up with 2 haploid cells. And so now meiosis 1 is complete, but the thing that's really important to notice here is that the sister chromatids are no longer identical to each other. We've introduced some degree of variation there. And now we're ready to begin meiosis 2, okay, because we have to pull those sister chromatids apart now. So enter prophase 2 now. In prophase 2, the microtubules begin to attach again to the duplicate chromosomes, and the chromosomes begin to move towards the equator once again, and it's during metaphase 2 that we now look a lot like metaphase in mitosis, where it looks like sister chromatids lining up on that equator, just like we're used to seeing. The difference is that these are not genetically identical to each other anymore because of that crossing over event that occurred. So during this stage is where we now begin to divide things up. Anaphase 2, we have now split those sister chromatids into separate independent chromosomes. And they begin to get pulled to the poles until we reach telophase 2. And in telophase 2, the chromosomes are now all being pulled to opposite ends of the cell. And the nuclear envelope begins to form around each set of chromosomes, just like we're used to seeing in mitosis. The cytoplasm begins to divide, and the result is 4 haploid cells. Now, that was a lot to cover in just a few slides here. So at this point, if you want to pause and watch those videos over again, now would be a really good time to do that. Okay? But I'm going to continue on. So, and then here's where you can watch the meiosis detailed video. How much variability is actually possible in offspring? Now, we actually have 3 different things that contribute to genetic variation in your offspring, in your babies. We already talked about the first one, right? The first one was in that crossing over event. Okay. Remember, in crossing over, we're taking some DNA from the mom DNA, the mom chromosome, and we're switching it with the dad DNA from the dad chromosome. And in so doing, we create 4 new and unique genetic complements. However, there's another place where variation can get introduced. During metaphase 1, when those homologous chromosomes were lining up on the equator, they lined up randomly. That is to say, after crossing over occurred and they started lining up on this equator, this one up here, this mostly purple chromosome over here, we happen to have it lining up close to this pole over here, while this one lines up with this pole down here. But it could just as easily have occurred where this one down here lined up, switched over, and was lining up with this pole. Now, why is that important? Well, because these are being pulled to poles along with other chromosomes. And what could happen is this chromosome right now is set up to be pulled to the poles along with this one here. But if they were switched, then this one would be combining with that one there, thus creating a brand new genetic complement. Now, in this process, you are creating lots and lots of variation. The last step in the random variation introduction of all of this is in the actual combination of sperm and egg as it gets fertilized. Each sperm is unique. Each egg is unique. And if by random chance one sperm and one egg combine, they're going to produce a whole new organism, a whole new offspring compared to some other sperm and some other egg. So that's why if you have 12 children, assuming they're not identical twins, then they are all going to look very similar to each other because they're all going to be some different combination of your DNA and your spouse's DNA, but they're not going to look identical. They're going to be some different combination. So this random alignment event where the chromosomes can line up either closest to this pole or this pole is actually very important. How many different combinations of chromosomes can we make? Well, you can actually figure this out. Initial contacts between the microtubules and the chromosomes can actually grab onto this one, or they can grab onto this one, and they can pull one to one side and this to the other side. But if these two were switched, it would set up a whole new combination here in the resulting sperm and eggs. So random alignment, it means that either the maternal or the paternal member of a homologous pair can end up on either side of the equator of a dividing cell at either pole and produce a whole different genetic complement of chromosomes. And this is really nice because it means that the chromosomes that are in each of the developing gametes are actually some sort of a mixture of chromosomes from the two parents. And that's really nice. It means that your children are actually receiving a mix of chromosomes that came from your mom and dad and the mother and father of your spouse, and they're all being jumbled and mixed together to produce this brand new mix. Okay, so what are the actual possibilities, the actual statistical chances of producing any one genetic combination? Well, it's actually relatively easy to figure out. The number of possible combinations can actually be figured out mathematically. It's 2 raised to however many chromosomes you have. In the case of humans, we have 23 pair of chromosomes. So what we would do is we would raise this 2 to the 23rd power. That's how many different random combinations you can make. So let me show you an example here. Let's assume that we only had 3 pairs of chromosomes. Of course, we have 23 pair, but let's assume we had 3. If we had 3, then this would be one combination, this one right here, where when you split them apart, you would end up with one sperm containing that and the other sperm containing this. But if it combined this way, then this would be what it would look like in your resulting sperm. If they resulted this way, then this is what the combination would be. And then, of course, this is another one. Now, don't forget that it's just as likely that you could have an exact mirror image of this, where all these light blue ones would be on the bottom and dark blue ones would be on the top. So I'm only showing you 4, but the actual answer is these 4 plus their mirror images for a total of 8. So if we took 2 and we raised it to the 3rd power, it would be 2 times 2 times 2. 2 times 2 is 4, 4 times 2 is 8. So when you have 3 chromosomes, you can produce 8 different combinations of these 3 pairs of chromosomes. Of course, and so take a look at the chromosomal recombination video. That will kind of help spell it out for you. So if 3 chromosomes, 3 pairs of chromosomes produces 8 combinations, then what about if we had 4 chromosomes? Well, that would be 2 times 2 times 2 times 2, which is 16. What about 6 chromosomes? That's 2 times 2 times 2 times 2 times 2 times 2, so 6 2's in a row, which is 64. All right, what about when you have 23 chromosomes? That's a huge number, and you can't really do it on your calculator. Although you can do it in Excel. You want to give it a try, you can try it out yourself. And I put up here times 2 because don't forget you have 2 pairs of these. You don't have 23 chromosomes, you have 23 pairs of chromosomes. The number is, yeah, it's huge. What is that number? It is 70 trillion. 368 billion, 744 million potential chromosome arrangements. In other words, ladies and gentlemen, you'd have to have this many babies before you ever arrived at 2 offspring that looked identical. In other words, it ain't ever going to happen. So this is another way that we can get lots and lots and lots of variation, is in this random assortment of alleles. So we've discussed crossing over as one way to get genetic variation, and we've talked about random alignment as the second way. And then, of course, that third way, which we're not going to really discuss today, is all of that variation that you put into the sperm and the egg. Now you're taking, at random, one sperm over here and one egg over there and putting them together to make some unique combination that would be very different from just randomly grabbing a second sperm and a second egg and throwing them together. Very unlikely that you're going to be grabbing the same genetic complement. Okay, now, when does this meiotic event actually happen in humans? Well, in females, you're actually born with all of the oocytes you're ever going to have. So you're essentially born with all of the eggs you're going to have. They're essentially just in an arrested state, so that once you reach puberty, then meiosis 2 begins to kick in. So meiosis 1 has already really happened in females by the time they're born, but it's just waiting until puberty hits for meiosis 2 to kick in. And so, yeah, remains in meiosis 1 until puberty, when meiosis 2 kicks in. So one oocyte, that is one egg, is one menstrual cycle. Every time you have your period in the month, that is when one egg gets released. So females essentially have a finite, fixed number of eggs that they're ever going to have in their lifetime. Some females have more, some have less, and when you run out of them, that's when you hit menopause. Some women hit menopause in their 50s, others hit menopause in their 40s, or even in their 30s, or sometimes even earlier. It's a little different for males. In males, well, by the time we hit puberty, males are making sperm all the time. You have a limitless supply of sperm that you're going to essentially make in your lifetime. When you consider how many trillions of sperm you're making on a weekly basis, the numbers are staggering. How many females males can potentially inseminate? I don't even want to think about it. It would be horrible. Anyway, okay, moving on. So let's compare mitosis and meiosis, shall we? In mitosis, so thinking back to before the midterm, mitosis, this functions as an asexual reproductive method. We use it for growth and repair of cells, like when your skin cells break because you cut yourself. Then those cells around it undergo asexual reproduction and repair itself by making new cells. It occurs in all the cells of your body known as the somatic cells, and somatic cells are everything that's not reproductively or related. And they are essentially producing genetic clones of each other. They're exact photocopies of their parent cells. Whereas in meiosis, the function is to undergo sexual reproduction, and this only occurs in germ cells, or sex cells, if you will. And the outcome are completely different offspring genetically. So meiosis is incredibly important for producing genetic variation. Mitosis is incredibly important for rapidly producing genetic clone cells to repair oneself or to make new red blood cells after your old ones have worn out, or whatever reason. I have a really nice contrasting video, so make sure that you check out this video. It allows you to compare pretty easily the difference between mitosis and meiosis. Now, I want to go through and just point out a few places where they are different and where they are similar. In prophase in mitosis, the homologous pairs actually don't interact with each other. So there's no crossing over going on. But in prophase 1 of meiosis, there is crossing over going on. That is to say, the homologous pairs actually zipper together. The sister chromatids of one combine with the sister chromatids of another, and they actually exchange genetic information. So prophase 1 and prophase of mitosis and prophase 1 of meiosis are different in this crossing over thing. And that would be a really good question for midterm 2. Compare and contrast prophase in mitosis and prophase 1 of meiosis. You would say, oh, hey, in prophase and mitosis there is no crossing over going on, but it does happen in meiosis in prophase 1. What about an anaphase? Well, anaphase 1 of meiosis, this is where the homologous chromosomes actually get separated from each other. But in anaphase in mitosis, in anaphase 2 of meiosis, this is where the sister chromatids are actually separated. So that's kind of different. And so what about the results of mitosis and meiosis? Well, in mitosis, you end up with 2 diploid cells, and they are essentially photocopies of each other. They're identical to that of the parents. But in meiosis, you end up with 4 haploid cells. 4 haploid cells. And each one differs from the parent and from one another. I just have a nice diagrammatic way to demonstrate this. Let's start with mitosis here on the right. We have all of our DNA in here. This is already, they've already duplicated themselves, so we have our sister chromatids in there. They've all lined up on the equator. All of those chromosomes have lined up on the equator. You've now pulled them apart in anaphase here, and you end up with an identical photocopy cell. These two are identical to each other, and they're also identical to this cell here, that is before the duplication of chromosomes has occurred. Now let's look at meiosis. In meiosis, we line up the homologous chromosomes on the equator. We allow crossing over to take place. We then pull the homologous chromosomes apart in the first cellular division. We then line these things up on the equator again and pull them apart in meiosis 2, arriving at 4 haploid cells. So make sure that you understand the differences between what's going on in meiosis and what's going on in mitosis. And you're going to really appreciate knowing this difference when we actually cover evolution. Because in evolution, it's all about sex. I know it's kind of weird to hear somebody say that, but sex in this case is the solution to just about everything. So go and tell that to all of your friends at the party Friday night. They'll think that you're crazy. Okay, one thing that we're not going to really touch on too much, but I do want you to read these sections in the textbook, are on genetic abnormalities. There's a whole series of them that are discussed in your book, and most of these genetic abnormalities result in death of the zygote. That is, you never really get much past cellular division or create an embryo because the DNA is kind of messed up too much. But I'm going to show you a couple of examples where the offspring are indeed viable. What you're looking at here is the case of Down syndrome. And Down syndrome is an error where chromosome number 21, which should normally just have, a pair, right? There should be a pair of chromosome 21s here. Well, in this case, somebody with Down syndrome, there was an error in the duplication of the DNA, and you end up with 3 chromosome number 21s. Hence the name, Down syndrome is also known as trisomy 21. Tri meaning 3, somy meaning bodies. So 3 bodies at the 21st chromosome position. And it is a debilitating genetic disorder that usually results in mental difficulties and physical challenges as well. I have 2 other examples here. This one here is called Turner's syndrome, and it is where somebody does not receive a two complements of the X chromosome, and then in fact they only end up with 1. This O here indicates that no chromosome is present there. This person just has 1, and they have somewhat external female features, but they are sterile, so they do not produce um, eggs successfully. Over here on this side, you end up with a situation called Klinefelter syndrome. In Klinefelter syndrome, this is where instead of having two X's or an X and a Y, an individual here ends up with two X's and a Y, which is just too much genetic signaling to produce a viable offspring, or I should say a reproductively viable offspring. In this condition where you have XXY, you are also sterile. Externally, there are somewhat female features on the top in that individuals with Klinefelter syndrome produce breasts, but they also still produce a penis and testes. Now, there is no sperm production going on in those testes, but at least the external morphology is there. So this now concludes our meiosis lecture. Go and check those videos online, and I will see you all in cyberspace.